This is the third and final part of perceiving motion. Okay, and another tricky aspect of what's happening, some of the complications really for perceiving motion is the aperture problem, that when viewing a smaller portion of a larger stimulus is going to lead to some misleading information about the direction of that moving stimulus. So we can think of it as looking through the aperture of a camera, where we can only see some small selective portion of a scene. In this case, we're talking about one particular object, a larger stimulus. We're just seeing a small part of that. And this is basically what is happening with those directionally sensitive neurons in the cortex. They are not those complex cells that we talked about in the visual cortex. They are not sufficient for detecting certain types of motion. And a really good way to visualize this is by using a pencil or something, getting your fingers so that you're creating this kind of aperture. Okay, and if you move the pencil from left to right, you see this motion from left to right. But also, if you're in the kind of the center of the pencil and you move it up and to the right, if you're looking through that little hole in your fingers, what's being seen there is simply movement to the right. And he's showing this again with someone going up the steps with a flagpole. So if we're having this particular aperture, or we're seeing this particular piece of the flagpole, these particular neurons that are responding to that movement, they're going to be responding to movement simply to the right. They're going to be missing the fact, because they can't see the whole stimulus, that this is actually moving up and to the right. So how do we solve the aperture problem? So to solve the aperture problem, the visual system does this in a number of ways. So first, remember those complex cells, those directionally sensitive neurons that we saw in the visual cortex, their information is going to get pooled together in the middle temporal cortex. So the middle temporal cortex, or MT, I mentioned this in the very first PowerPoint on motion, that this is one of those areas that's responding really to a direction of movement as well as the speed of that movement. Second way the visual system solves this problem is by having those end-stopped cells. If you remember the end-stopped cells in the visual cortex, they were responding to a line of a particular orientation, moving in a particular direction that was also a specific length and with the angle. But so they're really responding to the movement of the ends of objects. And so that information gets pooled as well into the middle temporal cortex, which tells us more specifically about, is this to the right or is this up and to the right when we put this all together. If we look at our brain, that basically shows some of these areas. So we're getting those directionally sensitive neurons, the complex cells, and the end-stop cells from V1, from the striate cortex, and that information is going to be pooled in the middle temporal area. And just so you know, so the publishers have here medial temporal area, it's more accurately discussed as the middle temporal area, this area MT that responds to movement. And I also mentioned, pretty sure at this point, the medial superior temporal area, or MST, which would be just superior to that, or just on top or above that region. I'm actually going to come back to, so he points out some other regions as just comparison, so we see where this is in the brain and compared to some of those areas of the inferotemporal cortex where we were doing our object recognition, and some of those regions that are responding to specific types of objects. But I'm also going to talk at some point about the superior temporal sulcus. So the sulcus, kind of that groove that's going in, that's in the superior temporal area. And I will discuss that when we talk about how we perceive the motion of the body, or really biological motion. If we look at the physiology of motion perception, one of the ways they have looked at this is by using these dot arrays. And so the research I'm going to be talking about comes from Newsom et al., 1989. where they were manipulating the degree of coherence, which is the degree to which those dots are moving in the same direction. And so if you look at A, we're really looking at dots are moving randomly, so the coherence is 0. They don't have any coherence with each other. In B, you can see that 50% of those dots are moving in the same direction. They're moving upwards. And then in C, 100%. We have 100% coherence as all of those dots are moving in the same direction. And that is kind of this background for the studies coming up. So they're looking at these dot arrays, and what they found was a direct relationship between, one, the monkey's ability to judge the direction of the moving dots. So with random, obviously with the coherence being random and 0, we're not going to be able to see that those are moving in the same direction. But as we get more and more coherence, we're going to be able to see, hey, those are moving in a certain direction, right? Even though there is some random movement around there. But they saw a direct relationship between that ability to judge the direction and then the response of a neuron. So doing this kind of single cell recording in the monkey's middle temporal cortex. So just as these examples, as dot coherence was at 0.8%, so very low, the monkey cannot judge the direction, and the neuron's responses at baseline. So basically spontaneous firing. As dot coherence gets to 12.8%, the monkey can judge the direction that the dots are going, and the neuron is firing faster than baseline, so we see neural response. They've also conducted research with monkeys looking at dot arrays, where they've done this microstimulation of particular cells in the middle temporal cortex. And so if they have the monkey looking at dots moving to the right, as in the picture, so no stimulation going on, the monkey perceives the dots moving to the right. If they have the dots moving to the right and they're stimulating such that those neurons that are responding to downward movement are stimulated, then the monkey sees the dots moving down and to the right. So they're still perceiving what's out in the world, but it's being added to these neurons that are perceiving downward motion, and they perceive this down and to the right. So as we get to perceiving biological motion, I wasn't sure that I actually had my brain on this slide here, but you can see the superior temporal sulcus being pointed out again here. I have a couple of links to start with. One is the Biomotion Lab that you can go and play with that if you want to, and make the person walk more happily, more sadly, and see how, even though these are basically just dots on a screen, you kind of have to see it to know what I'm talking about, but even though these are dots on a screen, that they are giving us a great deal of information about people and their gait. So this is really similar to a point light walker, and he gives some examples of some point light walkers in the book, and he shows one where what they often do is actually have people, and they put lights on their joints, and so there are about 12, maybe 12 lights, and it's on the joints so we can see at the elbows and the knees and the hips, so we can use just those lights, and they turn off the lights so they're in the complete dark, and all you can see is these 12 lights or so, and we can use those lights to tell not just that this is biological motion, that this is a person moving, but we can often tell exactly what the activity is. And so the second link there is some examples of point light walkers performing specific actions, and you can kind of see if you can guess. They're both, so the first one you play with, and you can play with that as long as you want to or as short as you want to. The second one's like 30 seconds, so a really quick little video. When we are looking at these point light walkers, and so looking at this biological motion, we see that activity increases in that superior temporal sulcus, the STS, which is specific to biological motion. That area activates also to looking in people's eyes. When you say hello or whatever, when you pass somebody on the street, it is also responding when we are looking to the faces and eyes to try and figure out this kind of what people are talking about. But it's reacting to these point light walkers, as is the fusiform face area, as is the prefrontal cortex, or the PFC. So this appears to be really a network of regions that are activated by something like biological motion. They've also shown, Grossman, 2005, did a study where she used transcranial magnetic stimulation, so TMS, and we can use this. This is basically putting a magnetic coil over someone's brain, and either we can stimulate activity in that brain region, or we can overstimulate activity in that brain region, basically knocking it out or disrupting its ability to help us. So with the disruption of the superior temporal sulcus, this disrupted people's ability to detect biological motion. So they would have these point light walkers, or basically they had some dots on a screen that were like the point light walkers, then were showing us some kind of biological motion, and those are couched within these other dots that are moving randomly. And normally, even with pretty many dots moving randomly, we can still pick out the biological motion. We need, I hope I say this in the right direction, fewer of those random dots. We need those to be fewer when we have this disruption of the superior temporal sulcus, suggesting this is really helping us to perceive biological motion. I'm including this table from page 188 as it is a nice synopsis of the brain regions that are involved with different aspects of perceiving motion. So the striate cortex, where we're really perceiving the motion across some particular small receptive field. So the complex cells and the end stop cells. Again, with that, we have that aperture problem where we can miss the exact direction of motion, but that's going to be pooled together in the middle temporal area. The middle temporal area, MT, is telling us about the direction and speed of objects in motion. The medial superior temporal area, get those in the right direction there, or MST, is helping us to process really our own movement within a scene. So the processing of the optic flow, locating moving objects, and then reaching for those moving objects. All of that is aided by MST. And then the superior temporal sulcus, or STS, is helping us to perceive biological motion, which I've mostly talked about people with the point light walkers, but this relates to animals as well.